Our next speaker is Jake Hoffman, who will be talking about his work on the genetic response to desiccation stress in female Aedes aegypti mosquitoes.
Okay.
Yeah, Jake.
So how's it going, guys? As Nancy just said, I'm going to be talking about genetic response to desiccation stress in females Aedes aegypti mosquitoes. So Aedes aegypti are a tropically distributed species of mosquitoes. They're considered to be anthropophilic, meaning that they live in urban environments and they primarily feed on humans. They also happen to be the primary vectors for both dengue and yellow fever, which combined affect more than 400 million people annually. Current estimates put dengue fever incidence at roughly 40% of the entire globe. So these are incredibly relevant to public health. Another aspect of my research is climate change. Climate change is happening. It's objectively bad. It's having a ton of it's having a ton of different effects. In addition to changing temperature, we're seeing water shortages, a loss of biodiversity, changes in species distribution. If you went to the talk on Tuesday, you heard about the viticultural implications of climate change. Although in relation to all these other things, it's not really anything to whine about. So now we're going to look at the public health relevance. So with climate change, this is a map of dengue fever distribution in 1990 and a projection for 2085. The theme here is more red in the future. And we don't like to see red on disease distribution maps because that indicates more disease. The question then becomes, how do we get these prediction maps? Currently, these prediction maps are based only on the distribution of Aedes currently and future climatic predictions. It doesn't take into account the capacity of these mosquitoes to adapt to their environments. So that is the broad scope of this research. The idea is to increase our understanding of stress responses in these mosquitoes so we can better predict their future distribution to combat these diseases. On a more focused level, I was doing qPCR and designing primers to look at the expression of 4 genes that have been shown to be implicated in desiccation stress. So, the first of these genes is frost. It was originally discovered in response to cold stress, and since cold and desiccation stress on a cellular basis are very similar stresses, it makes sense that there will be cross-utilization of this gene for those 2 stresses. It's a mucin-like protein that has been shown to be upregulated in response to desiccation stress. The next gene I was looking at is DSAT2, which is a fatty acid desaturase, which modifies the composition of these membrane fatty acids by introducing a double bond into single bonded carbon chains, and has been shown to be downregulated in response to desiccation. The next one is PepsiK, which is phosphoenol pyruvate carboxykinase, Gesundheit. It catalyzes the first step in gluconeogenesis and has been shown to be upregulated in response to desiccation. The last gene I was looking at was HSP70, which codes for a heat shock protein. Heat shock proteins are your general cellular response proteins, cellular stress response proteins, and have been shown to be upregulated in response to a myriad of different stresses, including desiccation. So, I was doing qPCR, and for those of you who have not experienced the veritable hell that is qPCR, I'm going to explain it to you. So PCR essentially works by amplifying specific gene segments. So you find a gene of interest and you amplify that one repeatedly multiple times so that at the end of this reaction, this chain reaction, you have a ton of that product that you can quantify. You choose the gene segment that you're interested in by designing specific primers that target that gene segment. By looking at the amount of product that we have at the end of this reaction, we can assign relative values to how much template DNA there was originally. So, to design primers is difficult occasionally, especially in the case of Aedes aegypti, because the genome is not very well annotated. So essentially what I did is I found gene sequences of the genes I was looking at in Drosophila, found the protein sequences and put them into this program called BLAST, which takes the protein sequence and looks for similar ones in the Aedes aegypti genome. We then find those gene sequences and design primers based on that. And those are the primers that I used. Now we've got to see whether or not these primers will actually do anything. So, to verify that these primers are working, there are a couple things we need to do. First, we need to make sure that it is amplifying efficiently and amplifying the segments that we expect it to based on concentration. So on the x-axis here we have concentration, and on the y-axis we have CT. CT is a measure of essentially of the abundance of DNA that there is. We set a threshold, and then based on how many cycles it takes that template to amplify past that threshold, we get that CT. So what you would expect to see is at higher concentrations, we have a lower CT because there was more original product to amplify. And for this is our control gene S7. We see that this is the case. We see that nice downward trend. Okay, the next way to make sure that these primers are working correctly is by doing a melt curve. Different segments of DNA will melt at different temperatures based on how long they are. And so if you look at all the different temperatures in your reaction, you can see if things are melting at one place or if there's multiple different products that are melting at different points. So you see, in this case, we have this one nice peak, indicating that we are only amplifying one thing. So based on this data, we can say that S7, our control gene, is working well. So now we're going to look at frost. This is the standard curve for frost, and it doesn't look anything like the standard curve we just saw. This is odd. It shows that over a concentration gradient, there's no real change in the amount of transcript that we're amplifying. This is an odd result, especially in regards to the melt curve, which indicates that we're amplifying one specific segment. We didn't really know what to make of frost, so we stopped using it, but we'll come back and talk about it later. So for PepCK, you can see that the standard curve was nice. However, the melt curve indicates that we're getting two different peaks, implying that either the primers are binding to multiple places within the DNA template, or that they are binding to themselves and amplifying themselves, which is called primer dimers. Either way, we can't get good data from a reaction that has a melt curve like this, so we stopped using it. You're just going to have to trust me when I say that DSAT2 and HSP70 both also worked. So we decided to verify the results we just saw. We ran the PCR products out on a gel. As you can see, DSAT2, HSP70, and S7 all had one nice band, indicating that one product was being amplified. Frost had a single band, but it was incredibly faint. And then PepCK over here had two bands, telling us exactly what I just told you, but it's nice to hear it twice. So for stressing these mosquitoes, we would raise mosquitoes in a happy mosquito environment, let them acclimate for a couple days, and then put them in these vials to stress for desiccation. So the vials were sealed with parafilm and had a dryerite desiccant added, creating a desiccating environment. We'd add 10 female mosquitoes per vial, so this exact setup except with 10 mosquitoes. And then we would leave them out for 4 or 8 hours with unstressed controls running simultaneously, so just without dryerite. At this point, we'd put them in the -80 freezer to kill them and store them, and run an mRNA extraction and do a cDNA synthesis, which takes forever and leaves us with a sample of template DNA that is representative of gene expression. So here are my results. On the x-axis are my two different stresses, and on the y-axis is the mRNA abundance relative to those untreated controls. So a full change of 1 indicates no change. Anything greater than 1 is an upregulation, and anything less than 1 is a downregulation. You won't see specific control samples presented here because those controls are inherent in getting these values. So for DSAT2, we saw an upregulation after both 4 and 8 hours, however with no statistical significance. And then for HSP70, we saw an upregulation after 4 hours and then a downregulation after 8 hours. This downregulation was so close to being statistically significant that we're going to talk about it anyway. So thinking about this doesn't necessarily make sense originally. You've got this protein that helps with the stress responses, but after a certain amount of stress, it just gets turned off. That doesn't seem like it would help the mosquito survive those longer stresses. So after looking in the literature, we actually figured out a way that this could work. So what you have the HSP70 DNA, which gets transcribed based on a stress into the RNA, which then gets translated into the protein. At lower concentrations of this protein, that's how it will remain. That is the system that will continue to have happen. However, once you get higher levels of protein, after 8 hours of desiccation stress where this is occurring, you see the cessation of transcription of this RNA. So in general cellular conditions, when you have RNA, it's around for a while, and RNA degradation forces come in and degrade it, so it decreases over time. For HSP70, however, it's been shown that under heat stress, this RNA degradation is turned off, allowing the RNA to stay around and continue to make protein. In my stress, however, we were doing desiccation stress, which does not have that effect, which could result in this downregulation of HSP70 RNA that we've been seeing. Okay, so now we're going to come back to frost. It was amplifying a specific segment of DNA, but was not doing it consistently based on concentration. The only way that we could think of to explain this was that it was being expressed in such incredibly low levels that the dilution made no difference, essentially indicating a loss of function of this gene. So essentially, it's evolution 101. You have a gene that responds to, and over time, it responds to a certain stress. And if the organism that this gene is in is having to deal with that stress, that gene will remain the same. If, however, in the case of Aedes aegypti, it is in an environment where that gene is not necessary, such as a cold response gene in a tropical mosquito, we can see that it changes into something else. So future directions for this piece, for this work, confirming the frost de-evolution would be interesting to see. We still have a lot of work that would need to be done. And increasing the resolution of the HSP70 response pattern would also be incredibly interesting, because over the summer we were doing just base desiccation resistance tests on these mosquitoes, and they can survive for longer than 8 hours. So it's not like this gene gets turned off and then they die. This gene gets turned off and they're surviving for far longer. So it would be interesting to see how that expression pattern would vary based on time. And then lastly, we could use, once this data is more refined, we could use this to make population comparisons. So these mosquitoes were derived from a population in Godiri right here. And once we have good expression patterns, we can then compare them to other different regions, Aedes aegypti and other different regions, to understand the evolutionary path that it could be taking under these various stresses. And with that, I'd like to thank all these people for their amazing amount of help, and I'll open it up to questions.
Questions for Jay?
So, is one possible reason that you see HSP70 go up and then down, that in a desiccation resistance thing, it is helping some of the proteins that are needed for desiccation to fold properly or to be present anyway. And then once you have them, you don't need it anymore. Whereas with heat, that's a little bit different. You constantly need it to help proteins fold in a hotter environment.
That could totally be possible. It would depend, I think, on how long-lived those desiccation proteins are, and that'd be an interesting thing to look at for sure. Yeah.
Did you flash freeze these mosquitoes? Because the first thing you told us was that cold stress was similar to desiccation, and then you killed them with cold.
Yeah, we stuck them in the -80 freezer, and since they're very small, that'll kill them pretty quickly.
How did you originally decide to embark on this project, and why?
Well, I was frantically looking for something to do for a thesis, and I emailed Emily, and she was like, "Yeah, you can work for me." And then after a lot of hard work, we came up with this.
So Jake, have you done any sequence comparisons with the Aedes aegypti frost and some closely related species to see if it's accumulated mutations?
Yeah, that was actually one of the ways I was thinking about, looking at frost and whether it had actually been lost, was looking at that. For sequence comparison of the proteins, for all of the proteins I did except for frost, sequence similarity was between 70 and 80%, but then for frost it was around 35%. And so doing some type of phylogenetic analysis based on evolutionary time to see if that's what would be expected if it was being a gene that was being conserved would be interesting to do.
I Thank you so much, Jay.
Right.